Hi, my name's Haley Blackwell, and this is my presentation, a deeper dive into the evidence behind the theory of endosymbiosis for the origin of eukaryotic cells. Introduction and question. The question that this project was based off of is there any evidence for the theory of endosymbiosis for the origin of eukaryotic cells? Now, starting off with the biologist who made the case for endosymbiosis was Lynn Margulis. He actually made it in the 1960s. Now, when he came up with the theory, a lot of scientists were very skeptical because no one was around a billion years ago to see the process of endosymbiosis. Now, going into endosymbiosis, a simple definition could be an organism living inside of another organism completely. Going on to the theory of endosymbiosis, it explains how organelles inside eukaryotic cells are descended from ancient times, kind of like a unicellular prokaryotic organism. Now, an example of endosymbiosis is the relationship between a termite and the microorganisms inside of its gut. Observational and comparative data are a relevant experiment. Personally, from the research I've done on the subject of endosymbiosis, I believe that a lot of the evidence comes from observational data, as it can be based on the mitochondria and chloroplast. Now, they both have their own DNA, and both of their DNA are similar to bacterial DNA. And this has all been proven true for RNA, DNA, ribosomes, chlorophyll, also known as chloroplast, and protein synthesis. On the next slide, there's more information. Going in deeper of the subject, essential observational and comparative results or experimental results. Starting off with membranes, the mitochondria, they have their own cell membranes, kind of like prokaryotic cells do. DNA, mitochondria have their own DNA genome, and DNA is passed to its offspring from a mitochondrion. Each mitochondrion that have their own DNA genome, they're kind of circular, like a bacterial genome. Now, when the DNA is passed from the mitochondrion to the offspring, it separates from the host cell genome and the nucleus. Both the membranes and the DNA can be shown in the picture in the left-hand corner. Moving on to reproduction. Now, in order to reproduce, mitochondria, they have to multiply by pinching in half. Now, bacteria also uses the same process. New mitochondrion must be produced from the parent mitochondrion, and they cannot build new ones if the cell's mitochondrion has been removed. Reproduction, as I said before, carries the same process, kind of like bacteria. Now, in conclusion, there's decades of work accumulated for this evidence, and because of the evidence to prove endosymbiosis, a lot of the scientist community actually now supports Margulis's ideas that the endosymbiosis is the best explanation for the evolution of eukaryotic cells. It not only applies to the mitochondria, but also other cellular organisms as well, starting from prokaryotes to eukaryotes that expanded to plants. A lot of scientists believe that endosymbiosis, that the event generated mitochondria, and this is because all eukaryotics have them. Scientists also believe that the similar event happened, bringing chloroplasts into some eukaryotic cells, which then started to create the lineage that led to plants. Now, the eukaryotic cell evolved more than 1 billion years ago, and the organelles now they're completely dependent on their host cells. An example could be many key proteins needed by the mitochondria are imported from the rest of the cells. In conclusion, the evidence suggests that these chloroplast organelles were once free-living bacteria. Because mitochondria are found in all eukaryotes, the endosymbiotic event that produced them have occurred early in their evolution of eukaryotes. Since then, these organelles have become completely reliant on their host cells. Thank you for letting me present today. Here is my references that I used for my presentation. Thank you.